Good morning. Can you all hear me in the room? Excellent. My name is Steve Webb. I'm the CEO of the Global Institute for Food Security, or GIFS. For those of us that are in the room present, there's coffee and mini muffins at the back, so make sure you grab some. And I'd like to welcome you here, for those that are in the room as well as those that are online, to today's seminar where we're going to discuss the opportunities and excitement around engineering biology. I want to begin by acknowledging that we are on Treaty 6 territory and homeland of the Métis. We pay our respects to the First Nation and Métis ancestors of this place and reaffirm our relationship with one another. I also want to acknowledge that I have a very nice, and yes, for all the people that said it's a new tie, it's a tie by an Aboriginal artist named Norman Cox. Norman Cox lives in the Kimberley region of Western Australia, and Australia is also going through the recognition and reconciliation process like we here are in Canada as well. And this is really important for all of us everywhere to recognize our First Nation and Métis ancestors of all the places that we're from. I'd like to thank everyone for taking time this morning to join us. At the Global Institute for Food Security, we take pride in being a connector and innovation catalyst within the agri-food ecosystem, and it's events like this one that we can do that. I'm looking forward to today's conversations that will occur in the Q&A sessions following both of this morning's presenters, as well as on the sidelines, because again, lots of great conversations happen when we're in person. We'll get to as many questions as we can today. For those online, please type them into the chat box and we will work through them. Your moderator, that's me, will sort through them and ask the panelists questions. For those that we're not able to answer, we will attempt to follow up with you as well. Again, we want to try to leave the room with more information and more knowledge around what is engineering biology and how it plays an important role in agriculture and food innovation. I think engineering biology has lots of different names, and we had our International Science Advisory Panel discussion earlier, and one of our panelists suggested that we call it accelerating biology. Engineering biology was advocated by other members of the group, and we also have discussed the concept of the original name, synthetic biology. What does all of that mean? It's really the combination of engineering principles such as automation and miniaturization, coupled with biology and computational tools. So A, B, C, to drive the design, build, test, learn cycle to allow us to do science at scale quickly. And again, our two speakers will talk about that today. It's an exciting opportunity, not from just a science perspective, but also from a business perspective. It represents a $2 to $4 trillion opportunity in the business space, and about a third of that is for agriculture and food. It represents the next industrial revolution, so the bio-revolution. At GIFS, we're proud to have taken a leadership position of this within Canada. We're a member of Canada's National Engineering Biology Steering Committee, and we are building out Canada's only engineering biology platform focused on the agriculture and food sector, and again, networking across other Canadian institutions. Before I begin, I want to take a few minutes to acknowledge some special guests. I've mentioned this before. Our special guests are our International Science Advisory Panel members that are present with us today. Margaret Gatsby is right there. She's the chair of our International Science Advisory Panel. Joerg Bohlmann, who is going to be speaking from the Michael Smith Labs at UBC, is right there. Herman Spangenberg, who is this afternoon's speaker for accelerated breeding. And Heiss, where's Heiss? See, Heiss is so tall. It should be easy for me to see him, and it's rare that I can't see him right away. Heiss is with Alberta Genome Alberta. Is that correct, Heiss? Thank you, because I keep transposing the names because Ontario has buggered me up. in terms of how they perform. And again, we've been lucky to have the 4 members of our science advisory panel here to visit us at GIFS. It's the first time they've had a chance to visit GIFS since we've moved to our new facility and have really been executing against our new strategic plan. A little bit about GIFS. We were established about 10 years ago. We're in our 11th year. We're a unique industry government partnership. Again, all 3 of the sectors that need to work together to spark invention and deliver market impacting innovation. It's a rare 3-way partnership, not the only one, but very rare. We're a global organization, not just by what we do in terms of the impact we have around the world, but most importantly, we're a global organization because of our about 80 staff. They come from 24 different countries. Our founding partners are Nutrien, the government of Saskatchewan, and the University of Saskatchewan. Our mission is to work with partners to discover, develop, and deliver innovative solutions for globally sustainable agriculture. And again, there's 3 parts to that. Partnerships, everything, innovation's a team sport, and we can't do it alone. And at GIFS, we want to partner with everyone, and we'll hear about partnership opportunities and networking through our engineering biology and accelerated breeding platforms later this afternoon. Discover, develop, and deliver. It means we're focused not only on creating knowledge, but also translating that knowledge into market impacting innovations. Because again, great ideas that never get to the market do not make a difference in terms of sustainability. And sustainability means economic, environment, and social. And here in Canada, particularly in agriculture and food, we had an opportunity yesterday to be at Ag in Motion and see how new innovations have delivered economic, environmental, and social outcomes and goals. It's not an or, it's an and. Our purpose is to be a connector and innovation catalyst. I mentioned that we do this by hosting events such as this, but also through access to our science, our people, our capabilities. We work from lab bench all the way to products in the field. And last night at our little GIFS barbecue informal night, we had commercial partners of ours in the room, along with academic and government partners. We serve as a connection bridge, again, linking and growing the pie. Yesterday, our ISAP members and some of our board had a chance to tour the Saskatchewan Food Development Center. And again, we work with the Saskatchewan Food Development Center as well as many other partners. I see Allison Ferry from NRC in the room, and I better put a plug in for NRC's partnership as well in our work that we're doing in Bangladesh, along with the University of Saskatchewan, the Global Institute for Water Security. And again, bringing these opportunities together to grow not only what we do here in Canada, but also making an impact internationally. At GIFS, we're organized and we've invested in the building of capabilities, capacity, and competency. Our people represent the competency, our capabilities is our facilities, and our capacity is our team's focus on scaling. And again, we've intentionally built cutting edge technology platforms. We're going to hear about our baby one, if you will. I think I've heard that from Nancy. The baby is engineering biology that we're just building now, but our most advanced is our sequencing capability and our data analytics lab and services that we offer in the community here, but around the world for sequencing and bioinformatics. And again, these are at scale capacities that enable research that we do in terms of accelerated breeding, biomanufacturing, and policy and regulation. But the intention of the double-edged arrows on the screen is they reach to our partners. And these programs and these platforms are designed to help our partners scale and drive their programs forward faster. What I want to talk about today a little bit is introduce our first speaker, Dr. Joerg Bohlmann. Dr. Bohlmann is a professor and distinguished university scholar in the Michael Smith Laboratories at the University of British Columbia in Vancouver. Just a little plug. My master's supervisor did a sabbatical under Michael Smith, and I had a chance to meet Michael Smith. That was the first time I'd ever met a Nobel Prize winner. And again, really important science contributions here in Canada. Joerg is a co-director of the UBC Genomics Science and Technology Graduate Program, as well as a holder of several prestigious fellowships and recipient of numerous awards and distinctions for his research and leadership. He's the one that I like the most is he's a member of our International Science Advisory Panel, and he's been a great mentor for Ben Scott, who's our engineering biology platform lead. And today, Joerg will share strategies and learnings from his work and research involving engineering biology, discuss opportunities and applications within this emerging field and on the leading edge of this science. And again, it's a real opportunity for me to welcome Joerg to present this morning. And Joerg, he's a walker, so he will not be on the podium apparently. And it'll make it much more entertaining than where I'm standing behind the podium. So Joerg, take it away.
Can everybody hear me in the back of the room? Wonderful. Thank you so much. And great. Thank you, Steve, for the wonderful introduction. Not so much for introduction for me, but really more importantly, the introduction to engineering biology. And I think I should just drop my talk and pick up all the keywords that Steve gave us, because there were so many important things that he mentioned, not the least to refer to Michael Smith, who 30 years ago was a pioneer in biotechnology. And he recognized the importance of bringing biology and engineers together, because he recognized that we learn to understand biology. And if we can engineer it, we can use it for human applications and make it better. And another point to pick up was the wonderful tours that we did in the last couple of days. Yesterday, we visited Ag in Motion, and then we went to Vido. And Vido is, of course, at the very forefront of engineering biology and things in the human health application and animal health application space. But we also went to Ag in Motion, and I was reminded of my time as a boy when I grew up on a farm with my uncle and my granddad, and I looked at the tools that we were using 40 years ago, 50 years ago. And I saw the tools that have been engineered for farming now. And just doing those comparisons, this is what engineering does. This is what engineering does for food production, for food security. Now, apply the same thinking to what engineering can do to biology, where Ben Scott is coming in with the platform that he's building up at GIFS, where we understand biology now to a level and can use the ingredients and components and modules in biology to engineer biological systems to harness all the potential of nature for human benefits. I will talk about one very specific example today. And what I want to do purposely, don't tell you the final and complete success story out of an academic lab, but almost the opposite. I will tell you what an academic research environment can accomplish with a PI who has somewhat limited funding, with graduate students and postdocs who do ingenious work, who are very smart, but also where in an academic environment, we do hit the roadblocks. And this is the roadblock, and Steve related to this, from discovery. In Canada has been fantastic in discovery in the space of natural products, biochemistry, translating this into applications to benefit humans on a scale. And what's missing there for this translation is exactly what GIFS is currently building up, an engineering biology platform. The type of technologies and the type of scaling that is not possible in an academic lab, but that is essential to come through this valley from discovery to innovation in, for example, corporate development, company development, and full application. The example that I will be talking about today relates to food security in a human health context. I'm talking about a bioproduct, a plant-based bioproduct. Compound is called montbretin. If you don't remember this name, just think about plants produce hundreds of thousands, maybe 1 million different, highly specialized metabolites. metabolites that plants do not need for growth and development, beyond carbohydrates, proteins, lipids. They produce this large plethora of specialized metabolites that we appreciate as humans every day. From your first experience in the mornings when you brush your teeth and you have menthol as a refreshing agent in your toothpaste, and you have your coffee and you appreciate caffeine and all the flavors and fragrances in your foods, all the way through. All these specialized metabolites are produced in nature through specialized biosynthetic systems that plants have evolved over hundreds of millions of years. Plants have done a fantastic job, but we know today that we can harness this potential of plants to produce these compounds, re-engineer them, redesign these biosynthetic systems in heterologous hosts such as yeast. And this is what Ben is doing, and make the same compounds to scale and makes them better and produce new to nature compounds. Again, I will talk only about one specific compound named Montbritin A, a compound that has the potential to be developed into a new treatment as a nutraceutical and or pharmaceutical for diabetes, specifically type 2 diabetes. And I'm putting this picture of a plant in the center here. This is the plant that produces this compound naturally. It's actually the only plant in nature that produces this compound. So it's a very, very rare metabolite. Here's the chemical structure of Montbritin A. You don't need to remember this. There will not be a quiz on what the chemical structure looks like. And I will look at this compound actually much more in terms of its building blocks, modules, and then how we can use the biosynthesis of plants to arrange these modules to produce an active compound. This plant is actually one that some of you may appreciate from your own gardens. It's a plant that is native to Southern and Eastern Africa, but is being grown all around the world today as a horticultural plant. It's not an agricultural crop, it's a horticultural plant. It's invasive in parts of the world. For example, in New Zealand, it grows beautifully in gardens on the West Coast. You may see it all over town in Portland right now, John, and it's being highly loved by hummingbirds. And if you find this talk entirely boring, you want to tune out, just think about why is it that hummingbirds who constantly suck on sugar water don't get overweight and don't get diabetes? Quiz question. So from here, I want to acknowledge Dr. Steve Withers, fantastic colleague in the chemistry department at UBC who introduced me to this molecule, Montbritin A, a molecule that he has been marveling about as a chemist and biochemist. And he came to us about 8 years ago with a critical question as to, I can't make this compound, but I need it. How does a plant actually produce it? How can a plant do something that a chemist cannot do? Here's the outline of my presentation. I'll talk a bit about diabetes. I talk a bit about Montbritin biosynthesis, how the plant produces it. And I talk about our opportunities to go into bioengineering. First, the challenges, how far we can go in an academic discovery lab, and then the opportunities where with working with a bioengineering platform such as GIFS, or GIFS specifically, I should say, such a compound can be explored for scaling it up to production levels. So diabetes, there's two different forms, type 1 diabetes, type 2 diabetes. Type 1 is the inability to produce insulin. Type 2 diabetes is insufficient use of insulin. Both lead to increased blood glucose level. And this has a whole range of comorbidities, damage to various organs, and is actually a very debilitating disease for many people around the world. Current numbers, and those were just published in a Lancet study in the end of June. Currently, there is, based on the Lancet study, 523 million people in the world affected by diabetes. So more than 500 million people. That's huge. The majority of these, over 90%, are type 2 diabetes, inefficient use of insulin. In Canada, and this is a number that is very much the number that applies across most of the world, despite the some uneven picture depicted on this map here, 10% of people live actively with diabetes. It's 4 million people. The projections are scary for diabetes. The Lancet study projects that in the next 25 years to 2050, the number of people affected by diabetes will rise to 1.3 billion people. That's more than 10% of people globally. That's a huge economic burden, health burden on individuals and communities. And because of this, the Lancet actually calls it a defining disease of the 21st century. Now, it doesn't get the publicity that cancer gets or that infectious diseases get because it is not your immediate killing disease. It's sort of the silent killer in society. And again, think about 1 in 10 people, potentially more in the future, affected by diabetes. It is among top 10 leading causes for premature death and disability. Huge economic cost associated with this. It's often associated with obesity or even being overweight. It's not an old people's disease, and it's not a rich country disease. It's affecting young people, children, people in their early lives whose careers and lives are being affected by it. It's present in all types of society, high, middle, low income countries, and high, middle, low income societies within countries. We often forget, just because we are probably a fairly privileged segment of the Canadian society, we have low income segments of the Canadian society that may be overrepresented on our diabetes statistics and are overrepresented. It's a central issue, therefore, for global food security. And if you read the latest, and not just the one latest, but the various UN food reports on food security, diabetes is a theme that runs through the entire report on global food security published by the United Nations. One may think, for someone of life privilege like me, that diabetes is preventable, entirely preventable, because it can be type 2 diabetes. There is a genetic predisposition for it, but it's also a lifestyle disease. Healthy diets, physical activity can prevent the onset of diabetes. So what we should actually avoid is unhealthy diets, sugar-rich foods, sugary beverages. We should all exercise frequently, avoid lack of physical activity, so lifestyle epidemic, leading to the issue of it's a lifestyle choice, right? We could all be very healthy, but that's very, very, very naive to say. That comes from a position of privilege to say it's just a lifestyle choice. We live in a world of lifestyle inequity. There's many people who do not have the choice between this being their nutrition during a given day or this being the nutrition in a given day. I have these choices. The person who drives a taxi 16 hours a day and needs to eat while drive, the single mom who works 2 jobs and puts 4 kids through school, she may not have the budget to put this on the table. So lifestyle inequity is actually a big component in this, often leading to poor nutrition, poor nutritional choices, forced choices. So the whole issue of diabetes, type 2 diabetes, is not one that is quickly going away just by everybody eating healthier and exercising more. We have this big issue of lifestyle inequity in our society and around the world. So healthy diets, physical activity would be one, but we still need to control diabetes with medication and ideally with nutraceuticals, things that we can actually put into our foods to make our foods healthier, specifically towards prevention of diabetes and controlling diabetes, ideally at the pre-diabetic stage. Now, what are current type 2 diabetics? The most sold type 2 treatments target starch digestion. We get most of our sugar intake, much of our sugar intake through food in the form of starch, starch-rich diets, be it your pasta, be it your rice, be it your bread contains starch. Starch is being digested in a 2-step process. it's being broken down by alpha amylases into so-called oligosaccharides, short sugar molecules, which are further being broken down by glucosidases into glucose molecules. The glucose is being resorbed into the bloodstream. And if you're type 2 diabetic, you get this rapid boost of your blood sugar level after a meal. Current treatments for type 2 diabetes inhibit the last step in starch degradation, the step that is catalyzed by alpha glucosidases that breaks down oligosaccharides into glucose, which leads to lack of production of glucose and, by co-effect, into the accumulation of oligosaccharides in our digestive system. So the oligosaccharides pass through our intestines and they become a formidable substrate for all sorts of microorganisms in our microbiome, gut microbiome, to ferment these oligosaccharides. They normally don't get to see them, but they get to see these oligosaccharides now and start fermenting them. And this causes a whole array of extremely unpleasant side effects for people who are on these medications, of which flatulence is just the mildest side effects, going all the way to heavy cramping, pains, diarrhea. And these very unpleasant side effects often lead to poor compliance. You may be on a prescription to control your type 2 diabetes with these drugs that inhibit the alpha glucosidases, but you will not take them because they're not, well, they come with these unpleasant side effects. So our colleague Steve Withers in the chemistry department at UBC, who I acknowledged before, set out together with his a group of colleagues at UBC to find an inhibitor for the first step in starch digestion, which are the alpha amylases that are being produced by the human pancreas, the human pancreatic alpha amylase, or abbreviated as HPA. So I may use this term HPA occasionally, human pancreatic alpha amylase. The idea was, if we can inhibit the first step in starch digestion, we never produce these nasty oligosaccharides that cause the unpleasant side effects, but we very much like retain starch, and starch is fairly inert for the gut microbiome. If anything, it may actually have a positive effect if it passes through the gut system and doesn't release the glucose, so it doesn't cause the sugar spike. So they set out trying to discover a compound that is highly specific for the inhibition of the first step in starch degradation, the human pancreatic alpha amylase. They published a study in Chem Biochem in 2008, where they showed the results of their work. They screened initially 30,000 natural product accessions and extended this discovery work to 50,000 different compounds. They got compounds from libraries that they had created and developed themselves, from going into plant natural products, marine natural products, fruit extracts, purified compounds. They bought commercially available library in their screen, high throughput screen for a compound that has a biological activity to specifically and selectively inhibit the HPA, the first step in starch gradation. They found just out of the 50,000 compound, one that had this very, very specific activity. So high throughput comes in here and translates further into what Ben is going to do, high throughput biology, high throughput engineering. They found this compound. For the biochemists in the room, the inhibitor constant is 8.1 nanomolar. That is very, very, very high affinity as an inhibitor for this compound. The chemical structure is complex, as I mentioned, and again, there is no quiz on what the chemical structure looks like. Think about it as an assembly of different Lego building blocks. And I had a conversation with Ben about how our children enjoy playing with Lego. And these are Lego blocks that nature uses. It uses this purple, blue, green, orange, and yellow compounds. Myristicin is a flavonoid structure. That's the core structure of this compound, MBA, and it decorates the plant that produces this compound, decorates this molecule with sugars that it adds to it, xylose, glucose, rhamnose, and it puts a so-called phenylpropanoid compound as a side chain on caffeic acid. So 7 different building blocks, and just try to play the game like 7 different compounds. There's many, many different ways how they can be arranged to be put together. Take your 7 different Lego building blocks and put them together, and how many different combinations can you create? What's important for the biological activity of this compound to function as an inhibitor of HPA, and therefore being relevant as a nutraceutical or pharmaceutical to treat type 2 diabetes, is this very, very specific configuration of this molecule. We found, and this is Steve Wizard's work and his group, that just a very, very minor modification on the caffeic acid side chain, just the difference of 1 oxygen atom, makes this molecule entirely inactive. And this comes back when we think about engineering biology strategies, how specific nature is and how synthetic chemistry cannot do this, what nature can do, and where we can use the catalyst from nature to rebuild and redesign these structures in heterologous host systems that we can actually engineer. So here was the opportunity. Steve and his group had discovered this wonderful compound. They had taken it through animal studies with diabetic rats. They had done the toxicity studies, came out very, very successful. The compound is now approved by Health Canada for phase 1 clinical trials that should be completed this year. And well, now build a company, produce this compound, license the technologies maybe to pharmaceuticals or companies in the food space, and voila. Well, there's a big, big, big problem that I mentioned before already: supply. This compound is extremely rare in nature. There's only 1 plant that produces this compound, and this plant decided to produce it in a place that is difficult to access. It produces it in bulb-like structures called corms below ground, and it only produces this compound in this particular place in very, very small amounts, and only during a very short time in the year. It accumulates for a period of time, but again, this is not a reliable source. This plant has never been taken into any larger scale production. It is being propagated in the Netherlands as a horticultural plant to provide bulbs for gardeners, but there's no agricultural technology or horticultural technology to plant and harvest this compound or the bulbs of this compound on any larger scale. So extremely rare in nature. Chemical synthesis is inefficient, and trust me, Steve Withers is a very, very good synthetic chemist, and they have tried it and it's not efficient. So this is where Steve came back to me, and he wanted to know, if I can't make this compound as a synthetic chemist, how is it possible that a plant can make it? And this is where we get back to appreciating actually the capacities of plants to produce things that humans can't make, but that we can harness now for engineering biology to make. And this is where the concept of metabolic bioengineering comes in. There's a whole lot of different things that Ben can make and build with his engineering biology platform. He will be able to make proteins, enzymes, ingredients with a wide range of applications. But what he can also do, he can make compounds like this in an engineered yeast system. But there's a bit of discovery work that needs to be done first. And this is what can be done in academic laboratories. What we cannot do is taking it to the high throughput engineering system. So this is what I told Steve how we would do this. We would eventually come to the bioengineered production system for this compound that makes us entirely independent of this plant that produces this compound in smallest amounts. But what we need to do first is we need to get our hands on all the genes and the enzymes that they encode to assemble this molecule in a very specific way from its 7 different building blocks, in a very specific way. And don't put an extra atom of oxygen in there, because that makes this molecule inactive. So using this highly specific and specialized metabolic capacity of a plant to build this compound by discovering the genes and enzymes. How do we get to these genes and enzymes in a plant that has never had any biochemical or genetic or genomic studies done on it? Well, we apply large-scale metabolite profiling and large-scale transcriptome profiling, do correlations across these 2 different discovery spaces, metabolome and transcriptome, and correlate which genes are active when and where in the plant and are likely candidates for the biosynthesis. This would be the very detailed discovery strategy that we applied. Again, we start with metabolites and we come to pathway reconstruction in a heterologous host and then need to take it to the bioengineering. Just as an aside, there was no genomics, genetics, biochemistry work done in the system before. There were no available chemical standards. This was all a typical academic type of discovery, where the high throughput component was metabolomics and genomics. And the bioinformatics. Bioinformatics, very important, bringing information on metabolites and genes together. Over a period of just a very few years, we were able to discover the complete biosynthetic systems, the 6 different genes and enzymes that are responsible for assembling this molecule from its starter molecule by addition of individual side chains to make this very specific compounds. And being discovery research, we published this work, so it's all out there. We capitalized heavily in our discovery strategy on the very fact that the plant produces this compound specifically in the corms during a short specific time of the year. So we did correlation studies, which genes are active at which time in the corms and not in other places and times of the plant, correlated metabolite data and members of different gene families that are catalytically active for producing these type of compounds. And we're now at the stage where, yes, we have discovered the complete biosynthetic pathway. We have all the genes in hand, and now can we reconstruct and can we bioengineer the system. In our lab, we could take it to the level of proof of concept of reconstructing this biosynthetic system that is in nature unique to Montbrechia plants, reconstructed in alternative hosts that can be grown in greenhouse facilities or that can be grown in a fermenter. But we don't have the capacities to take this through an engineering approach to scale, to optimize it. I give this away already. We continue to end up with very small yields only. So what are our options for developing a production system for this compound now? Well, one is we could consider crop development, but I won't even go there because there are so many. No, it won't work because of A, B, C, D, and many, many different reasons. So here's the fantastic opportunity for bioengineering. We've been exploring 2 different systems. One is a relative of tobacco plants. It's actually not tobacco. It's Nicotiana benthamiana, a relative that is native to Australia, that is being used widely now in the plant biotech industry to engineer biosynthetic systems for the production of therapeutic proteins, diagnostic proteins, but also specialized metabolites. And here, yeast bioengineering. And this is where Ben is taking this work from us and taking it to the next level. I hope he will. Very quickly, what we've accomplished and why we've been working with the plant as an engineered host. It is a plant. It produces all the upstream metabolic building blocks for this compound, so we can capitalize on this. It has the proper plant cellular compartmentation. It can grow, compared to other plants, this Nicotiana benthamiana plant can be grown in greenhouse facilities relatively rapidly to produce biomass, not as fast as a yeast fermenter can grow though. And we have a collaboration with Plantform, an Ontario-based plant biotech company, where we're actually exploring this. So that's underway, metabolic engineering in yeast. We've hit a few roadblocks. One of the roadblocks is that because the advantage of a plant is that it produces all the metabolic precursors, it also produces a lot of metabolic precursors that this molecule incorporates now that we don't want to have incorporated. So it does not only produce MBA, the compound that we want, it also produces related compounds called MBB and MBC that are entirely not active. So, and it produces actually more of those compounds. That's a challenge when we go with metabolic engineering from plant into plant, that we suddenly engineer in our target metabolic system. This target metabolic system is now supplemented with the endogenous metabolic system of the plant. and they build us a lot of side products that we don't want. So that's a big challenge with plant metabolic engineering. And this is what we are pursuing now to overcome these challenges with plant form. Yeast has challenges, but yeast also has the opportunity. The challenge of yeast is it does not naturally produce the precursor building blocks, but that's also the beauty of it, because we have a clear slate. We can put a plant metabolic pathway into yeast and don't need to be bothered with what are all the possible side products that we don't want to get. We can focus on this. We need to do more engineering, and because we need to do more engineering, we need an engineering biology platform for this, not just an academic lab where students clone genes more like at 10 genes at a time and reconstruct them in 10 sets of 10s, but the large scale. So we need to engineer the capacity into yeast to produce the core flavonoid myristicin compound, the side chain that puts the precursors for the various different sugar and phenylpropanoid side chains. And those come from these precursor biosynthetic systems that I'm drawing out here that the plant produces, for which we have all the genes now that we can engineer into yeast. What we've accomplished so far, and this is at the academic lab level, we've engineered modules for these 4 different capacities into yeast to produce a flavonol precursor, phenylpropanoid precursor, sugar precursors, and the assemblies of the MBA. But this is separate engineering, different modules in different yeast strains. And this is establishing proof of concepts that all of these genes actually do work in yeast. This is not yet taking it to scale. What we need to do now, and that is the big challenge, but it's a possibility to do. It's a realistic challenge. It's not an impossible one. 20 years ago, this would have been an impossible thing to do. Now, this is something that with engineering biology, we can do. Engineer the complete biosynthetic system into yeast and bring the production level that we can achieve in yeast up 100-fold or 1,000-fold to come to levels of production that are relevant for a commercially viable production system. This is just showing a result that proof of principle in yeast by engineering different modules into yeast, that yeast can produce this compound. And this is very much where I want to lead this as an example into Ben's work with an engineering biology platform, where he would come in with larger scale design, build, test, learn, an optimization process. And Ben will talk about this, how in the capacity that he is building at GIFS, a large number, and we're not talking about tens, we're talking about thousands of gene combinations can be optimized through this cyclic process of design, test, build, test, and learn. Large number of different gene combinations from all of these different genes that we need to engineer into yeast, that we can engineer into yeast. Large number of different gene variants. Just we have discovered what the native plant uses to produce these compounds. Individual catalysts along the way can be optimized. So create gene variants that are better. Just the same way as I saw on Ag in Motion yesterday. The combine that my granddad used is not the combine anymore that is being used on the farm now. So we can actually engineer things to make them better. This is where Ben comes in. Large number of different promoters to drive the expression of these pathways. Is this all dream? No, this is reality. Things like this have been done in other spaces, mostly focusing on the pharmaceutical space, but this is a space that I'm talking about here that's at the interface between human health and food security. Diabetes is a food security issue. There is studies that have been published mostly out of laboratories in the Bay Area, Berkeley, and an associated company, Amyris, that has developed an anti-malaria, or that has developed an engineered yeast system for the production of artemisinin, an anti-malaria treatment. Recently, the same group out of the University of Berkeley and collaborators around the world published an engineered yeast system expressing more than 20 genes to produce the anti-cancer drug vincristine, or vinblastine, sorry. This is a compound that is derived from Madagascar periwinkle. I choose these examples just to tell you that these things are possible. In those environments, academics have been working with bioengineering platforms or engineering biology platforms for quite some time, for the last 10 years or longer. And it required the engineering biology platforms to take it from discovery. Curiously, much of the discovery work that is behind artemisinin biosynthesis and vinblastine biosynthesis is Canadian discovery. But it's being actually developed through engineering biology somewhere else, because up until now, we've been missing such a platform in the Canadian ecosystem. There's Canadian scientists who did the pioneering work on artemisinin biosynthesis. Pat Cavallo, who was a scientist at PBI in Saskatchewan for many years. Dae-Kyun Roh, who is a faculty at the University of Alberta. Canada's Vincenzo De Luca, who worked at PBI early in his career, did much of the discovery of genes in vinblastine biosynthesis. So we don't want to export Canadian discoveries always. We need to have these technology platforms here. And I think this is just one more reminder: diabetes is not a small disease. It's a huge disease. It's a big issue for global food security. And acknowledging all the people in my lab who've been involved with this work as graduate students, undergraduate students, postdocs, technicians. The acknowledgement that most of this work was done at the University of British Columbia, which is on the ancestral, traditional, and unceded territories of the Musqueam, Squamish, and Tsleil-Waututh people. So the very important acknowledgement of our First Nations and Indigenous people. And still the question: why is the hummingbird not getting diabetic, although feeding on sugar water? So from here, I think I lead it into Ben's presentation.
So Joerg, thank you very much. I think we got a few minutes, and again, the clock here is always at midnight, so we'll be able to manage that. The one thing that I wanted to kind of take away from your presentation is you mentioned it again, the linkage between health and food security. And it's a conversation last night that I had with Margaret about thinking about food security in terms of nutrition. And again, the linkage of not just calories, but the quality of the calories to be able to address these issues. I think the intersection between plants, between food, between health, again, initiatives here at the University of Saskatchewan, like the One Health Initiative, are interesting, but also all over the world. And one of the institutes that's similar to GIFS, which is a government academic industry partnership that I was on the board of, the Indiana Biosciences Research Institute, is focused on cardiovascular and metabolic disease. And its number one disease is diabetes. And again, the opportunity for partnerships, collaborations, and the convergence, not only of technologies, but of the disciplines that we have in abundance here. And Joerg, thanks for your comments about the tours yesterday and the AIM and the Vito and the Food Center. But again, that convergence, and Andrew is right there from Vito, that again, the opportunity for us to be thinking bigger. And again, thanks for setting up the opportunity. Again, that idea of translating knowledge into an innovation or a development, and then into an actual product that can make a difference in folks' lives. We have a few minutes for questions. Margaret.
In its host plant. And the reason I'm wondering is just, is any of this function of interest to something like potato? Would it be an advancement?
That's a great question, Mark. And I did not tell you have to ask this question. So potatoes, I come to this in a second. What is the natural function? We don't know. We have been speculating on it. And there is some ideas for actually testing our ideas about this. Many of these so-called specialized metabolites that we find in nature, be it flavor compounds, aroma compounds, fruit pigments, have actually functions in nature of a plant interacting with other organisms, attracting pollinators by colors, attracting fruit dispersing animals, including humans, also by color, by flavor, and aroma. So these are positive interactions. This compound may well function in its natural environment, in the plant that produces it, to protect the corms against corm-feeding herbivores, small rodents, for example, that during the non-vegetative period of the year, while the corm is a storage site for starch, would feed on it. These animals would feed on it, but they would get no uptake of sugar from this molecule. So can we engineer this into potato, perhaps? Potato protection? I don't know. That's a side question. But this would be what we currently speculate or hypothesize is the natural function, an undefeated compound. And animals learn. Rats learn very quickly and rodents learn very quickly. If I feed on this and I don't get energy out of this, I feed on something else. It may also come associated with a bitter taste. Don't eat it again.
Great. We do have a question from online. It relates, first of all, the person online wants to say, great presentation, Joerg. It was really insightful. And one of the question really relates to, with an engineering biology approach, do you see an opportunity to develop a structure activity relationship around the lead molecule and further improve the potential of the inhibitor?
Yeah, absolutely. I mean, structure-function relationship has been done by Steve Withers to the point of co-crystallizing this metabolite MBA with the amylase. They found what the essential components are. They learned why the related compounds that are just different by one oxygen or one methyl group are inactive. But with this space is wide open now. And again, to use engineering biology platform to test are different glucosyltransferases active on this? Can we make compounds that are similar, but perhaps even more active or easier to produce, right? Yes, absolutely. Yeah, this whole space of structure-function relationship becomes much more accessible on a larger scale with engineering biology techniques.
Raju, but wait for the microphone because we missed the first part of Margaret's question.
Very nice talk, Joerg. In the heterologous system used for engineering this complex biosynthetic pathway, there will be several intermediates that will be produced during the process. All those intermediates influence yeast growth and regulation. Anything is known?
No. And this is again where the working with an engineering biology platform comes in to test this. Ideally, we would not need to feed any intermediates or precursors, but the yeast would produce them all themselves. But again, this is what we're testing in much larger numbers comes in.
Yeah. Another question related, can you bypass some of those metabolites by feeding later phase metabolite into yeast? So you can bypass if there are so many negative effects.
Skip some of the early steps in the biosynthesis. Some yes, others not. Some are commercially not available or would be extremely expensive. So the UDP activated sugars are very expensive to purchase. The CoA esters of the phenylpropanoids are not available or very expensive to purchase. And yeah, use the enzymes that nature is giving us and engineer them into heterologous host like yeast.
Great. Other questions? Nancy?
Thank you, Dr. Bohlmann. At first, when I saw the flower picture, at first I thought it was a prairie lily, and I thought about what are the possibilities of prairie lily natural products. But I'm more interested in your realization that what your vision of, sort of world without type 2 diabetes and getting this drug into the hands of the end users, that realization that what you could accomplish in your academic lab, the vision wasn't possible, and how you then sort of came to that realization that scale of the research needed that next step and phase, and maybe just talk through that a little bit.
Yeah, thank you very much. It's a very, very important question because it's both a question of frustration and excitement. So the excitement comes from, yes, with genomic technologies and metabolic technologies, we discovered a metabolic pathway system that 10 years ago or 20 years ago would have taken perhaps 20 years to discover. We could do this in just a few years and get the publications out and enable postdoctoral fellows to move on with their own careers and setting up their own labs now. The frustration is now we need to take it to the next step. And now we need to test not just 10 different combinations of genes in yeast strains that we can engineer at the bench in the laboratory, but now we need to scale this up to test thousands of combinations, make hundreds, tens or hundreds of strains every day. And this is where in a typical academic lab, we run into a brick wall. And this is why historically, many of these discoveries didn't lead into the full-scale applications to benefit humanity. And it was partly that engineers like Jay Kiesling and his many colleagues in the Bay Area who come from an engineering, from a chemical and biological engineering background, went back to look at what did natural products chemists and biochemists do 20 years ago. And said, well, now we can actually use these tools and these modules that biology provides us. But again, in Canada, we don't have these type of capacities yet. There's one very beautiful foundry that with Dr. Vince Martin started at Concordia. It's different from what is being built at GIFS. And this is exactly where, coming from the excitement of the discovery through the frustration running into a brick wall, the next excitement comes now. Yes, we can do this.
Terrific. Hey, I want to thank everybody for questions from Joerg. And again, there'll be some time, I hope, at the end for some more follow-up afterwards, but we do have to get to Ben, our next speaker. So Joerg, thank you so much. And just a quick introduction for Ben. Ben is 6 months, Ben, with GIFS. So he's not even a year old in GIFS years, so to speak. Ben came to us after he completed his postdoc at the National Institute of Standards and Technology in the United States, working at their microbial biofoundry in Maryland. And he also was the business development lead, and Joerg introduced Vincent Martin's foundry in Concordia. And that's where Ben was before joining us. Ben his roles have given him really a strong appreciation for the automation and how it can expand scale and scope of engineering. And today, Ben's going to talk about, Dr. Scott is going to talk about engineering biology and its connection to food security and explain what about Biofoundry is and share our vision for our new engineering biology platform. Ben, come on up. All right, come on down. You're the next contestant.
Okay, thanks, Steve, for the intro and for Joerg for the really beautiful, insightful presentation setting me up here. So we have some big targets globally that we sort of set for us. So in under 30 years, we're going to need to increase our food production, and globally, this is the royal we need to increase our global food production by 56%. Which is a pretty big goal. In addition to that, if we're serious about climate change, as we all are, and we want to meet this 2-degree climate goal that we've all heard about in the news, we need to accomplish this goal without using more land and also while reducing emissions. So really ambitious goals that we've set for ourselves. And in addition to this demand, we're already seeing some growing food costs. So at this point in time, well, as of May, Canadian food prices are increasing faster than at any point in the last 40 years. And I know we've all experienced this directly going to the supermarket, and we will talk to our families about how ridiculous this is. But this likely isn't going to get much better anytime soon as we're faced with the global demands for food. And the climate goals that we set for ourselves. So kind of bleak, but I like this quote because it means that we really have no choice but to embark on this greener revolution globally And it's not all doom and gloom. There's technologies, there's ways of meeting these challenges that I want to introduce to you today. One of those ways that I think we can meet these challenges is through engineering biology. There's probably thousands, hundreds of thousands of different definitions, different ways, different arguments for defining engineering biology. But this one's my favorite one because I can explain it at Thanksgiving to my family about what I do. And it's using biology to build useful products. It's something that anyone can really understand. The opportunity that Steve mentioned at the beginning here is massive from an economic point of view. So $3.6 trillion for a global market in the next 20 years, which is incredibly large. It's 10 times the size of the laptop and PC market that many of us own. And it's growing very fast, so 3 times faster than the robotics market globally. And food and ag is a very significant part of this. So there's a huge economic opportunity in the food and ag space, equivalent to the human health space. And as Joerg pointed out, these are not mutually exclusive. And we can think of this really as the same sector. There's also some very interesting applications in materials and energy and consumer goods. So how do we approach biology as an engineering platform? This is one of my favorite kind of analogies, I think, because computers have become so ingrained in our society and culture. We're used to thinking about computers. Biology can be thought of in an engineering context very similarly, and we can engineer at any one of these scales. So they're not mutually exclusive. In fact, in biology, they're all connected for the most part. And we can choose certain scales to fit the purpose of the engineering goal. In addition to that, and something that's unique to engineering biology, is that the scale up of how we actually produce something is also an engineering goal. Computer chips can be more or less manufactured the same way, whether you're making 1 or 1 million. In biology, that's not true. So there's another challenge, another layer to how we can engineer biology as well. There's also some really unique aspects about biology that lend itself to engineering. They can be challenges, but they, I think, can also be opportunities. So there's it's self-replicating. It has an ability to self-repair. And for me, I think it's easiest to imagine a car doing any one of these things and how kind of ridiculous it would be to imagine that. But biology can do this. We can also randomly introduce functional diversity and then select for function. Again, in using the idea of a car, it would be like you open up the hood and you just sort of like start taking a hammer to it and just throw a bunch of screws or something into your car and hope that it's going to start working. In biology, something could evolve to actually do that and improve the function. So sort of humorous to think about, but it's true. Extremely compact, so thinking about millions and billions of cells that in a test tube, each one in a different system that we can select and evolve, can sense and respond to the environment. And I think the last one I wanted to end on, because of the huge potential that it has for the climate emergency, is that we can store and then repurpose carbon. So this means that we could take CO2 out of the air or take a byproduct, a waste product that we would normally get rid of, and repurpose it into something valuable. So that's where, you know, the market-driven sort of forces can take over and really work together to achieve these ambitious goals. So some real applications, Joerg highlighted a number of them, but I wanted to highlight the fact, or point out the fact, that this is not a new field anymore. It's a mature and maturing field, and that there's applications already impacting people and making a difference worldwide. One of which is cancer immunotherapy. So Emily Whitehead was given CAR T-cell therapy. I think it's now 11 years ago, and she's proudly cancer-free since then. So really incredible impact that cancer immunotherapy is making. This was not planned that Joerg and I would both be talking about this, but this is one of the gold standard examples of metabolic engineering, and this idea of months and variability in growing a product and taking those genes from a plant and putting them into yeast. And so you get down to weeks, very precise control of the product that you want. And then starting to build into the food discussion, you can go and buy plant burgers, meat-free burgers at the supermarket now, that use heme that's derived from yeast instead of from animals. And heme is a critical flavoring and coloring agent that gives them this real meat flavor and color. And this is already, you can buy this on the shelves now. And I think it's wonderful because it really does mean that this product is completely animal free for consumers. So those are some examples broadly in engineering biology. I wanted to drill down to some more specific examples for global food security, which is the whole purpose of setting up this work here at GIFS. And I started with thinking about what the end product is. And because there's almost too many different applications, I wanted to start with sort of what they could actually look like, the things, the tangible things that could reach the market. So this could be a plant, a microbe, a protein or enzyme, or a small molecule. And there's sort of 4 different categories here. I'm not going to go through, I was going to explain each of these, but I'm just going to sort of highlight the fact that there's many different examples and touch on a couple of these. There's many, I'm going to just skip through to here. Okay, so there's many of these that can be products at some scales, some at all of these scales, and really just highlighting the fact that there's many different engineering biology applications in food security. This is one that I really wanted to highlight because of its potential and just so many different things that it can impact. So the sustainable and scalable production of novel ingredients, flavors, micronutrients, medicines. Maybe I'll just wait, there's some audio issues. Anyway, ingredients, flavors, micronutrients, medicines, pesticides, pheromones, antimicrobials. This idea of growing something versus extracting it or using fossil fuels to manufacture it. And this covers all of these different scales. Okay. There we go. Okay, and then the last but not least on here is the upcycling and valorization of waste products and biomass, which I also touched on previously, but it's this idea of taking something that we would ordinarily be probably paying a company to get rid of, and instead using that as biomass, carbon, nitrogen, phosphorus, all the things that go into life, and creating a product out of it. And that's something that that biology can accomplish. And to feed into all of these different products, we can also think about the DNA, RNA, proteins, strains, microbes that go into those, and those can be products themselves. So at any one of these scales, we can be engineering biology for food security. Because it's such a great idea, there's lots of competition out there, and I wanted to acknowledge that and also build up the case for why Canada needs to invest in this and build this capacity. I was told about this just recently. I think it's a really interesting goal that Singapore has set for themselves to produce 30% of their domestic food needs domestically by 2030. So very ambitious goal, and they've invested very heavily into engineering biology as a way to contribute towards that goal. Our friends and competitors across the world as well have invested in used very significant federal investments in engineering biology, dating back now over 10 years, in the hundreds of millions of dollars that they put towards this. I encourage you to check out any one of these, as they're very ambitious, very interesting platforms that the Australia, UK, and US have set up. About 2 years ago, I made the case that we can meet some of our commitments to sustainable development while also making a real impact in supporting economic recovery from the pandemic. And you can go and read this paper if you're interested in getting to the nitty-gritty, but we argued that we can meet or certainly contribute to 11 out of the 17 UN Sustainable Development Goals using engineering biology. Yeah, Canada can really stand to benefit from this. There have been some interesting policy papers that have been put out in the last couple of years, so there is some traction for this field, and I wanted to acknowledge that. But policy papers aren't really enough. We need dedicated investment in this. So what's Canada's opportunity? The biomass. So something that really attracted me. I'm coming from a health sciences field originally and protein engineering. But one thing that I really like about agriculture and the field itself is that it functions on the scale of millions of tons of biomass. There's really no other part of the Canadian economy that does that. So when we think about feedstocks, biology, carbon sources, Canadian agriculture is a huge opportunity there. There's also biotalent. So Canadian universities produce some of the best trained individuals in the world. And Canada has very innovation-friendly policies surrounding gene editing, entrepreneurship, as well as immigration, because it's as fast and as many as people that we can train in Canada. Canada is still a relatively small talent pool just by the nature of the size of their population. And so we need to have innovation-friendly policies for immigration as well, which we do, which is a real opportunity that Canada has. And then last but not least is the biomanufacturing. So I really want to leverage the interest and investment that the COVID pandemic built in biomanufacturing and the health applications, but we can build on that even further. So the expertise and infrastructure that the federal government has supported through this can lend itself very well to what we want to accomplish in engineering biology as well. But there's a missing link, and this isn't just in Canada. This is true in research in general. Joerg set this up very nicely, that Canada is very good, and that the starting stages of this, we make significant investments, made great discoveries, but to then translate that into a product, a company, private sector, there's this gap where, yes, we've found MBA, but how do we produce it? And how do we turn that into a company? And that's where GIFS actually has been set up to play in. And the platform that I'm building is being set up to play in as well. Some other things that make biology particularly challenging, and I'm building up to the why we need to build a foundry to begin with, or a platform to support this, is that biology exists in a multifactorial space when we're engineering something. So that requires many accurate and also many different types of measurements when we want to engineer something. So you can imagine taking measurements across any one of these axes, and that's going to, and your engineering goal will maybe be on one of those axes. So let's say a particular temperature that you need something to function at, or a particular growth rate, but it may not be with the nutrients or the strain that you need or the titer that you want to produce. So that it's a very sort of slippery slope across any one of those axes. And as soon as you want to think about adding another axis of measurement to that, or another capability that your product needs, it quickly becomes difficult to measure just due to the scale. And then the function in biology is versus the DNA sequence that encodes that function is not linear. So you can imagine you start out where that little squiggly line is, and you want to get to the top right where that blue circle is, where that's the function that you need to create your product. Maybe that's the titer that you need, or the stability that you need, or X in biological function. The changes that we make in the genome of something, the actual DNA sequence that encodes for that, very small changes can result in completely different function. So it's almost impossible to predict how we would get from that starting point to the end point, even if we know where what we know that endpoint may look like. We don't know what the sequence is that encodes for that. Some more specific examples about why we need high throughput measurements and a platform to accomplish this. So let's think about a food enzyme. There's lots of enzymes that are used in food manufacturing and production. Where would we start in improving this? What changes would we need to make? Are there other proteins that maybe would have been better from the start instead of the one that we started with, the one protein? Or could we forget about nature altogether and think about what's possible beyond nature and design an enzyme altogether from scratch with desired properties? So any one of the answers to any of one of those questions may be out there, but we need high throughput measurements and ways of actually making educated guesses to answer those questions. And again, this wasn't entirely set up to support Joerg, but it's the same problem of a biomanufacturing of medicine, which is what strain, media type, culturing conditions are optimal, what gene clusters, promoters, gene copy numbers will give the best titer. And then with all of that data, can we then use it to make educated guesses and inform the next steps for a completely different product, a completely different goal? And again, the data that's required to answer any one of these questions is huge, and the measurements and the number of measurements are very large. So finally, we arrive at biofoundries. So biofoundries, it's a one place where automation and analytics infrastructure exists to support the engineering of biological systems beyond the manual scale. So to actually drill down and answer those questions that I've just posed. There's this great policy paper actually from the Australian government that put out this figure where a biofoundry enables us to go through the design, build, test, and learn cycle in one place and iteratively to engineer biology. And I'll bring up this design, build, test, and learn a couple more times because it's so foundational to engineering biology. So biofoundries enable us to iterate through this cycle bigger, faster, and bolder. It also, I think, I really want to challenge people to rethink biology because of what this capability enables. So we should get used to thinking at the scale of thousands, millions, billions of different evolving systems. And it's almost as if you're looking up at the night sky and you see all of those stars above you, and you're actually able to visit all of them and determine, well, maybe that planet actually is habitable, and making those types of scales of measurements. And obviously this allows us to ask questions beyond what's possible using traditional methods. And further to that, we need large and very good quality data sets to inform machine learning, and then use that data to save time the next time we want to engineer a system. And for all the hype, for all the promise of AI and machine learning, there's no getting around this requirement for having very large and also very good quality data sets to inform these models. So I'm going to highlight two of the biofoundries that I've worked at to illustrate the fact this is not science fiction and that we can build this. It's possible. So the biofoundry at NIST was set up to support test and learn along that design, build, test, and learn cycle. And they've published on this a few times now where they're able to measure the function of over 60,000 biosensors in one experiment, and then apply machine learning from that data set to say, okay, based on all of this, the collective data from all of these 60,000 biosensors, what mutations can we now introduce to get this exact function in the biosensor? And it appears to work. The other biofoundry that I've worked at is at Concordia University in Montreal, which was brought up. It was led by, is led by Vince Martin. And it's build and test focused, and it's capable of producing or building over 1,000 genetic constructs in under 2 weeks, and then test them in mammalian cells. So for any of you who've worked with mammalian cells before, this should really excite you because of just how challenging those cells are to work with. So here at GIFS, we're engineering biology to build useful products, which will enhance agri-food and natural products research via laboratory automation, achieving this scale beyond what's possible manually. And there's sort of three sandboxes that I've been envisioning the foundry to support, at least initially. Protein engineering, which is my bread and butter. That's the majority of my training. Natural products, which Joerg introduced. So this idea of a chemical of significance from a natural source and taking those genes out and putting them into something that's easier to grow and manufacture. And then biomanufacturing, which is once we have that strain, that yeast strain, that E. coli strain, maybe it's cell-free or a plant cell, how do we make more of it? And how do we make that scalable? Which was one of those challenges in engineering biology that I introduced at the beginning, because the function of biology at one scale does not always translate to a much larger scale at an industrial scale. These are some of the more specific examples. So with regards to protein engineering, I'm personally very interested in food and agricultural enzymes because they're already on the market, but many of them are naturally derived and they are not necessarily fit for purpose. So we can make improvements to them. Improved protein ingredients has been getting a lot of traction as well. So that includes improved nutritional properties, improved texture, flavor, and then the manufacturing of them as well. Joerg introduced pathway domestication, so taking these genes from a plant and putting them into yeast, as well as the metabolite characterization. So it's some way of high throughput screening of what's actually being produced at the end of the day. And then, of course, with biomanufacturing, strain domestication improvement and bioprocess optimization to get more of what we want. So these are the three themes. We've got some projects in mind, and I'd love to hear, maybe you've got some ideas for projects as well that would take advantage of this. So we need equipment to support this. And I did this first from the themes to projects point of view. So thinking about, okay, these are the goals, these are the engineering goals. What equipment do we need specifically to accomplish those goals versus the other way around, which is to buy the latest and greatest and hope that it's going to accomplish your goal. And then the specific equipment, I'm not really going to talk much about that today, many of which because we haven't actually purchased it yet, but we have ideas about what we need in order to accomplish these goals. But these suites is what I wanted to talk about today and introduce this idea of DNA assembly, microbial growth and phenotyping, and proteomics and metabolomics, and the types of scales that I want to work at. So with suite 1, DNA assembly, the goal or the whole purpose of it is to be able to assemble hundreds of genetic constructs a day, and then to verify their successful assembly. So we'll be looking at PCR fragments, plasmids, gene clusters, and synthetic chromosomes, and then doing some quality control on the production of these. Suite 2 is to easily and reproducibly culture aerobic organisms with thousands of microvolume cultures in parallel, so that we can actually pose those questions that would ordinarily be impossible at the manual level. And this would be with minimal human intervention or completely automated workflows. So this covers really the full gambit of taking DNA, putting it into a cell, growing it, and getting some type of initial read on its phenotype, but also from the microliter to liter scale with some benchtop fermentation capacity as well. So that from the microliter scale right to the liter scale. And I wanted to acknowledge that USask has made some pretty significant investments in getting beyond the liter scale. So this is a purposeful investment that we're making here at GIFS to not replicate that, but to work together to then take that beyond 1 liter. So what sort of scales are possible? I did introduce this idea of millions, billions. These sound really interesting, but I did want to say or point out the fact that it's possible. So DNA sequencing right now is the most highest throughput measurement for biology. So we can get, and these numbers are rough, but we can be comfortable with numbers around the million to billions of reads of a particular DNA sequence. And if we wanted to measure the abundance of a particular sequence in a mixture, we would need around 100 of the DNA sequencing reads to get some value that you're comfortable with. So based on that, and bear with me because it's a bit of a stretch, but if we can tie the desired function of something, and therefore the abundance is tied to the desired function. So if something has the function that you want, it's going to be growing better, so then it's going to be more prevalent in a mixture. We can then measure the function of 10,000 to 10 million proteins, pathways, strains in a single experiment. And these are not made up numbers. This is what they were doing at NIST with biosensors, and it's all by DNA sequencing as a readout, a function. Unfortunately, or fortunately, not all biological function can be tied to growth. And in particular, the titer of a desired product. So it would be wonderful if I could design a yeast strain and say, well, the one that produces the most MBA is the one that grows the fastest. But it's not that easy. And in fact, usually it's quite the opposite because of the metabolic burden that we impose on organisms. So there's a missing part to this test piece for this biofoundry. And that's where the last suite comes in, which is the metabolomics and proteomics suite, which is the quantification and analyses of metabolites and proteins at a scale of thousands a day. And that's 1,000 samples, not 1,000 metabolites, 1,000 samples a day. So we're building out high throughput sample prep, HPLC and LCMS capabilities, which will support the extraction from plants, microbes, and environmental samples. And I'm not doing this alone. And I wanted to highlight that the engineering biology platform that we're building is one of 5 platforms at GIFS, and that I'll be working very closely with the other platforms, and in particular 2 of them, which I'm going to describe very briefly here. The Omics and Precision Agriculture Laboratory, OPAL, it's a world-class DNA sequencing and genomics facility with workflows and technology that span all of the major short read and long read technology. So when I first visited GIFS, I was given a tour of the lab, and I got to see the latest and greatest sequencing machines from the latest and greatest vendors lined up right beside one another. And I was told, well, my lab's going to be right next door, and that you'll be working very closely with Austin to use this. So I was very excited as a scientist, and I can say we'll be working very closely with Opal on the DNA sequencing side. And then in addition to that, so when we approach these big questions and this big data, we need a way of analyzing this, and that's personally beyond my capabilities. I can do the wet lab, I can do the automation, I can generate the data, but to make sense of it all and to make to close that full loop, the learn part of the design, build, test, learn, I'm going to be working with data management and analytics, DMA, who is another platform here at GIFS, who has expertise in data analytics, machine learning to concisely summarize, interpret, and learn from this big data. And they've been already sort of hand in glove with Opal on the genomics level. So they're already very comfortable with biological data sets and thinking about answering some of these questions. So that's the full cycle of design, build, test, and learn that we're envisioning. I did a lot of hype, but I wanted some dose of realism because I've just started. I'm just here 6 months as of not even. It'll be like 4 days from now, be 6 months. So we're right here, and we want to be sustainable. We want to be self-sustainable and bringing enough clients or partners and interest to support ourselves. And I'm just buying the equipment right now. But I have thought through what we need to do to get there, and I do want to make progress up this hill every day. Some maybe a step back or a step forward and a step back, but I do want to make progress up this hill every day. And I need help doing it. This is more to look at the camera now and say, come apply, because there'll be people who aren't here right now, likely. But I'm hiring for a laboratory automation engineer, as well as a laboratory technician that will actually be shared with Opal, which I think I'm very interested and excited about because of this way that we're going to be working together. So apply now. I know this is likely being recorded, and maybe by the time it's up, the job postings will have closed because I want to hire pretty soon. So make an effort if you're interested to reach out. I'm happy to talk about this, or you can check out the job ads now. So this is the only purpose-built biofoundry for food and agriculture product development that's not just in Canada, but worldwide. We'll have access to state-of-the-art, and anyone can access the state-of-the-art equipment and expertise spanning this full cycle, with close integration with genomics and data analytics platforms, enabling and really drilling down to answering those questions and giving us next steps. And that's really to accomplish this goal, which is the high throughput measurements to bridge the gap between discovery and commercialization. So contact us today, contact me, you can learn more. And if you got an idea for a product along one of those 4 or 3 subject matters, I'd love to hear from you. Thank you.
So Ben, we're going to have to share the microphone.
Okay, great.
So again, for folks online, just type in your questions into the chat box and we'll get to it. For those in the room, put your hand up and we'll get the microphone, because I did not get that notice out when Joerg spoke. So Heiss.
Thank you, Ben, for this fabulous presentation. I think you made a great case that it's realistic to think that you can overcome the technical barriers associated with the production of these new products. But I think probably the biggest barrier to determine whether or not you're going to be successful may not be technical. It may be sort of regulatory and consumer acceptance. So how are you building those components into the building of your platform and the selection of the products or enzymes that you want to focus on going forward?
Thanks. That's a great question, Heiss. Thank you. The sort of 2 ways. One, a little bit more immediate in the selection of the products. So one, my interest in agricultural enzymes has been sort of formed around that question, because these enzymes are biodegradable. They are not a GMO, and we don't intend initially, or we don't have plans for introducing them into a microbe or a plant. So they exist as a product themselves. And if we were to use them, for example, for enhancing a crop or for making food production more sustainable or faster, they're not something that anyone would consume at the end. So that's some way that we can start to introduce this idea of engineering biology and improvement. That's not a direct consumer product. It would never, be eaten or consumed. The other is, I think, education and policy, and introducing this idea of engineering biology as a solution to problems that we all want to address and answer. I think given the choice between a product that comes from an animal versus not, if the products are identical, I think many people would choose the one that doesn't come from an animal. And if that product was made with engineering biology, with an ingredient that enhances that flavor properties and even nutrition, I think that becomes a lot easier for people to understand.
We have a couple questions from online. One is related to the build out of the platform that we're planning at GIFS. Question related to what scale is the fermentation capability going to be at GIFS? You alluded to it a little bit in your presentation, but can you elaborate a bit more, Ben?
Yeah, thanks, Steve. Right now, we're thinking of benchtop scale. Not a stainless steel steam in place fermenter, but a benchtop scale. That is informed by sort of two decisions. One is that we don't have enough space at the very present time to think about steam in place or larger scale than a liter to maybe 5 liters. But it's because, and I mentioned this, that we don't want to duplicate efforts being made at the food center or GAP or Vito for that matter, and the type of scale that they're already developing. So I can say up to a liter is planned, and that's the microliter to liter scale fully, and with bioreactors, not just a shaking flask, but a bioreactor up to a liter. But right now, we don't have plans for more, but given demand, maybe. But I'd really like to just work with people who are already building that capacity.
Lots of questions online, which is good. Any other questions in the room before I? Hi, Madavi. Is your microphone on?
With regarding to the fermentation facility, I think food sciences already have a facility to grow 100 to 200 liters. I think they have a facility already developed there.
Yeah, I got to tour the food center yesterday for the first time, and learn from Puba about what they're developing there. I think they've got a facility. I'm not going to quote numbers because they're going to be wrong, but they're building a much larger facility as well, which they're going to be commissioning soon, I believe. Yeah, thank you.
So there's 2 other questions on. Well, there's 3 other questions online. First one, I will answer it, Ben, because the question is, are we open to collaborating with other people? The answer is yes. We're very open to collaborating. Again, it's right in our mission statement about we work with partners. And again, the idea that innovation is a team sport. And not just in the ag and food space, because as Joerg mentioned, projects like MBA require access to technology platforms that can offer that transition to bridge from knowledge creation to impact in the marketplace. So yeah, we're open for business at GIFS, and we want to be your partner. And that's the bread and butter of how we work. Again, it's not GIFS, it's our partners, and GIFS supports our partners to get to where we all need to go. My good friend, Matt Noseworthy, asks a great question about how big a protein and protein space can you design and build in terms of this kind of platform and capability.
Oh, that's a great question. Okay, where do I start? I think when I go, it's with phage display. So phage display is kind of the smallest scale that we can think about. Maybe mRNA display, but 10 to the 9 or 10 to the 14, those sort of crazy numbers. So billion to, I don't know how many quadrillion that is, but a lot in a test tube. Getting functional information from those is very dependent on what the protein is. So that's not trivial. So when we think about phage display, the reason why they're so powerful is that they're protein binders. But if it's not a protein binding question, if it's an enzyme that's producing a product, you need some type of readout. So that becomes a little bit more challenging. Other ways to think about it are what exists in nature and genome mining. So there's almost unlimited diversity in nature that we could approach, or designing from scratch. And there's been some really interesting work in the protein design space very recently. It's almost a paper a week. David Baker out west has been leading this field, kind of built the field from scratch. And a recent paper, they created, I think it was about 8,000 enzymes using a computer, none of which exist in nature. And of those 8,000, 2 worked. But that's still 2 more than ever has existed in nature. So it's also possible to think at those scales as well. And I think that those numbers, that 8,000 is very quickly going to become 80,000 that we can manufacture, and then 800,000 and so on. So it's almost unlimited as far as scale. Yeah.
Okay, another question from online, but since I'm the moderator, an editorial comment. I think one of the things that both Joerg and Ben have introduced the concept, not just of big numbers, and I can test 50, like your colleague, Stephen, the chemist, tested 50,000 to find one active. It's the ability to apply statistical design to answering the questions, the ability to approach the question or test the hypothesis with a multifactorial design that's beyond the reach of a grad student. And this is really the application of science. It's not, I'm going to throw 100,000 darts at a board. This is a designed approach and science-based approach to be able to get to where we need to go. That's beyond the reach, was beyond the reach of me as a grad student, and every one of us as a grad student, because it's the integration of all of these technologies, and the opportunity to explore and build these spaces is really kind of cool. Other question from from online is, you mentioned aerobic. And Ben, any plans on going anaerobic? Not you, but
Or our platform.
no.
Yeah, not me. No, at this point right now, no. The aerobic, sort of a function of what's available as far as toolkits, genetic toolkits and characterized microbes. It's also challenging to work with anaerobes for obvious reasons. We would need to encase all of our robots in plastic and have, you know, hoods and gloves that we would reach into. There is a biofoundry or not a full biofoundry, but automated liquid handler set up at the Biozone at U of T that are already equipped for anaerobes. So maybe that person would be interested in reaching out to them, but we're not making plans on that. What I can say though is we're not limited to microbes. I'm very interested in plant cell culture and single cell plant culture as well, which I'm told function similarly to aerobic microbes, but have the machinery of plants. And we can grow and culture them very similarly. So I'm very interested in that. So it's not just yeast, E. coli, the sort of typical lab strains. And something I didn't mention as well is that thinking about a strain as a product, we shouldn't limit ourselves to what we're already comfortable working with. I'm very interested in characterizing natural diversity, going to those microbial repositories, testing what's in there, and building systems out using non-domesticated microbes as well. And there's a lot. There's many out there that are beyond what we're currently working with.
Thanks, Ben. Just got a comment online, and it's from our colleagues at the Food Center. They're building 2 trains with up to 10,000 liter capacity, just to kind of fully frame the investment. Vito has a manufacturing capability, again, healthcare focused, but again, the ability to actually produce product for the human and animal space. So again, there's a lot of growth, and how we fit in is we complement, we do not compete. And again, how we fit into the ecosystem is really, really important. Want to thank both of our speakers today. And again, I think Joerg did a wonderful job to talk about, to introduce the idea of convergence. And also, I'll be honest with you, once you get older, you start to realize you've bumped into all of these people more and more than once. Like Pat Cavello, I met when I first started my career here in Saskatoon. And then I met Pat Cavello again when he was working with Amyris. And Patrick Westphal, who was at Amyris, then worked with me at Dow AgriSciences, who we built an engineering biology platform as one of the co-authors on the Artemisen paper in Nature. And again, it's kind of this small world space, the Michael Smith Labs. And again, I think the idea of engineering biology is capturing all of the opportunity that we've invested in over the last 20 to 30 years around our genomic sequencing capability. I'm looking at Andy in the back here, and being able to think about how do we begin to integrate it. And the fundamental piece in this is engineering, biology, and the computer and machine learning. And that, again, is absolutely key of a teamwork approach and a synergistic approach. So first of all, thanks everybody for coming for today. There's a little bit of time. We're here until like 11 o'clock. And thanks to everybody for being online, and great questions. And if you have other questions, because normally like me, it'll be like, darn, I wish I'd asked Joerg that. I have the benefit of knowing Joerg's email, and I can send him the question. But if you have other ideas, please come and email them into the chat that's on the system now. We'll leave it open. And again, please partner with us at GIFS. We're open to partnerships and collaborations. And again, together, we can solve the big challenges that we're facing. Thank you very much.